My dear students, you are welcome. In our lesson today, this is a revision about calculations. We deal with some problems about percentage increase and percentage decrease. Okay, let's start. The first question here, first problem from paper 4, variant 3, October, November 23. Question number 4, number 6, CI. Okay, let's start. Table 6.1 shows some data about stomatal density in the leaves. Stomatal density means the number of stomata per surface area of leaf. How many stomata found in certain surface area of a leaf. Okay. In the leaves of one plant that is not xerophyte and 3 xerophyte plants. You know well about xerophyte means desert plant. Okay, not xerophyte means mesophyte means normal plant like oak tree. Here four species, oak tree and 3 other xerophytic plants. Okay. Um, in this table, we found number of stomata per squared millimeter on both upper leaf surface and the lower leaf surface. Here, in these figures, okay, the question about using the information in table 6.1, estimate the total number of stomata in an ice plant leaf اللي هو last leaf أو last species ice plant leaf with a lower leaf surface area of 8 square centimeters. Let's start. As you know before, here is the number of stomata per surface area. Surface area means squared units. Squared units means squared millimeter, square centimeter, squared kilometer, whatever, okay? But must be squared. Two-dimensional means squared units. For here, in this example or this problem, you ask about total stomata, total number of stomata per 8 in found in 8 squared centimeter. Look, please, as you know before, in your ruler, we found divisions, large divisions called centimeter. Each centimeter equal 10 units called millimeters, 10 millimeters. So 10 millimeters equal 1 centimeter. Okay. Now, how to convert centimeter to millimeter? Centimeter is large unit for length. To be converted into millimeter, we have to multiply times 10, times 10, okay? 1 centimeter equal 10 millimeters, okay? What about converting area units? Converting area units. As you know, area consists of square units, So we need to square all our lenses, say from squared centimeter to squared millimeter, we have to multiply. Instead of 10, we multiply by 100. 10 to the power 2. 10 to the power 2. Okay? And here, from smallest unit to larger one, okay? We have to divide from small to large, we divide. From large to small, we multiply. Okay? Now, look, we have to multiply 10 to the power 2 equal 100. Return to our question here, ice plant. Per squared millimeter, we found 42 stomata. Okay, so in 8 squared centimeter, we have to convert firstly square centimeter into millimeter by multiply 100. So 8 square centimeter times 100 squared millimeter equal 800 squared millimeter. It's okay. In each squared millimeter, we found 42 stomata. So the total number of stomata equal 800 times 42 equals this figure 33,600 stomata. It's okay? Okay? Okay. Now, another question from paper 4, variant 2, February, March 19, question number 1, C, single I. Table 1.1 shows the concentrations of four solids, four material dissolved in blood plasma. Here, in the blood, in the renal artery, in the fluid, in the kidney tubule, here means filtrate of glomerulus after filtration, here in the urine itself. Okay, 3 solutions, okay, and the 4 solutes dissolve in these 3 solutions: solute glucose, protein, salts, urea. Okay, solute concentration, the unit gram per liter per cubic decimeter in 3 parameters: in blood, in fluid, in kidney tubule, in urine. Look, in this figure, this our excretory system or renal system, urinary system, consists of 2 kidneys, left and right kidneys, 2 ureters, 1 bladder, and urethra. Okay, and their associated blood supply. Each kidney receive what? Receive blood. Here, look. Each kidney receive blood from renal artery. It's branch from aorta. Okay, branch from aorta. And also, the kidney after filtration of blood plasma, the blood after filtration released or removed from kidney, exit from kidney through renal vein. This bluish one and this one. Renal vein pour their bloods in inferior vena cava. Okay, towards the heart. Okay, let's see something important also. Second figure, nephron. Here the nephron where glomerulus, where filtration of blood plasma takes place. Glomerulus. Okay, this first part of the nephron, this yellowish tubule with different thickness, called what? Nephron tubule or kidney tubules. Okay, kidney tubules. This collecting duct collects urine from a lot of nephrons. So, to remind ourselves with these figures to answer this problem correctly. Look, calculate the percentage increase. He asked now about percentage increase in the concentration of urea. Urea is waste between the blood in the renal artery, where here in this figure, okay, and the urine, and inside the urine. Okay, show your working. For percentage increase, this formula, we have to remind ourselves with this formula, this good formula to answer this type of problems. Percentage increase equal final minus initial reading divide initial times 100 to give percentage. Okay, look please. Where is final? Final in urine, 20 gram per liter. Initial means in renal artery. So the difference divides the initial times 100. So 20 minus 0.2 divide 0.2 times 100 equal 900%. 900% is a great percentage of waste excreted every minute from our body. Thanks for God. Thanks for our kidneys to doing this great job. Okay. Third question about paper 4 also, October, November 20, question 3, DII. Doctors wanted to investigate the effect of exercise on the excretion of waste. They collected urine from people before and after running a long distance on a hot day. The results of their investigation are shown in Table 3.2. Their findings: average volume of urea per cubic centimeter, average concentration of sodium in urine per millimole per liter before running and after running. Okay, what about the question? Calculate the percentage decrease, not increase. Last problem about percentage increase. Here, percentage decrease in the average sodium concentration. So he concentrate on the lower room, okay? Sodium concentration after running compared with before running. Give your answer to one significant figure. Space for working. Average concentration of sodium in urine, millimole per liter, before running equal 85.6. After running decrease into 78.2. So there is difference. Difference divide initial means percentage decrease. Log please. Percentage decrease final minus initial divide initial. Where is final? This one. This after running. Okay. Where is initial? This one. 85.6. So final minus initial divide initial times 100. So the figure 78.2 minus 85.6 divide 85.6 times 100 equal this figure. Ooh. This figure with more decimals. Okay. But here, look, try to concentrate in your problem correctly, okay? To avoid losing any mark in your exam. Give your answer to one significant figure. So we have to round to make one significant figure means 9% minus 9% or 9% decrease, okay? Look now to this very, very pretty figure about some blood cells in blood sample. Look, this coming from variant 3. Okay. Variant 3, May, June 14, question 4, D single I. Look, this RBCs, red blood cell, red blood cell. This lymphocyte produce antibodies. Okay, phagocyte engulf microbes. And notice it has looped nucleus larger than lymphocyte. The lymphocyte is the smallest one. Lymphocyte nearly, okay, more or less like the size of red blood cell with large round nucleus. It produces antibodies through protein synthesis in their cytoplasm. Look also to this type of white blood cell, monocyte, not in our syllabus, but in this type of phagocyte also, called macrophage, very, very large, but its name coming during engulfing microbes in tissues, not in blood. In blood called monocyte, in tissue called macrophage, okay? No problem. Now look please to lymphocytes, and phagocytes are white blood cells. A woman had some blood tests taken before and during bacterial infection. Before infection with bacteria and during infection of bacteria. Okay. Table 4.2 shows the number of white blood cells in 2 blood samples. She gave 2 blood samples to medical lab before infection and during infection. White blood cell lymphocytes, sorry, phagocytes. Mean number of cells per cubic, here volume, cubic millimeter of blood. Okay, each cubic millimeter of blood before infection contains 1,300 lymphocytes, also contains 2,000 phagocytes. During infection, it contains 3,500 lymphocytes and 7,500 lymphocytes. Look, phagocytes, sorry. Look please to this part. This phagocyte, large in size with looped nucleus with granules containing enzymes, okay, like enzymes of acrosomes of sperm to digest microbe, okay. Here lymphocyte small, its size like the size of red blood cell nearly, with round nucleus. Calculate the percentage increase lymphocytes that occur during bacterial infection. Calculate percentage increase. This question about percentage increase lymphocyte. Where is the lymphocyte? Here lymphocytes. Percentage increase difference. Final minus initial divide initial. Okay. Here times 100 to give percent. Look also this during infection, this after or final reading, this initial reading. The difference divide initial times 100. Now compensate. Okay. This figure coming in your calculator. So we have to make another side of our problem. We have answers to the nearest whole number. So no need for this decimal points. Okay. So the answer 169%. Okay, the difference between before infection and after infection for the percentage increase of lymphocyte. Variant 3, okay, sorry, paper 3, variant 2, October, November 15, question 2, B single I. Look, here the lactic acid concentration in the blood of athlete was measured at intervals. At the end of the slow run, the lactic acid concentration had increased by 30%. During race, this race was slow run, okay, before competition, before the race itself, may be doing certain slow run. Then after that, during the real competition, they ran with fast speed. So here at rest, after a rest of slow run, the athlete ran at much faster speed on the treadmill. This actual race. At the beginning of this exercise, the lactic acid concentration in his blood was 100 milligram per liter. After 11 minutes running at the faster speed, okay, sprint, his lactic acid concentration was 700 milligram per liter. Calculate the percentage increase in lactic acid concentration at the end of the faster run. Show your working. Look, please. Also the same formula. Final minus initial divide initial times 100. Okay, same figure or same equation. Look, this before exercise, at beginning of exercise, here during the speed or after running for faster speed for about 11 minutes. Final 270 minus 100 divide 100 times 100 equal what? Equals this figure. 170% increase. Guess which one produces more lactic acid? Fast run or slow run? In the figure here, 170% increase in lactic acid during marathon, during real competition. What about slow run? Only lactic acid increases by 30%, only 30% increase. Okay, so more exercising, more running, more and faster exercising releases more lactic acid in our muscles to bloodstream. It's okay. Here, variant 3, paper 3, October, November 15, question 2, letter C. Look, here blood samples were taken from each of blood vessels J, K, L, and M 2 hours after a meal of rice. Table 2.1 shows the concentration of glucose in these blood samples. Table 2.1, Here's the 4 blood vessels and the blood glucose concentration per milligram per 100 cubic centimeter of blood. Look at this figure. Calculate the percentage increase in blood glucose concentration between blood vessel J and compared with blood vessel L, J and L. Okay, J and L. Express your answer to the nearest whole number. Increase, percentage increase final minus initial, divide initial times 100. It's okay? This initial, this final between blood vessel J and what L? L increased blood glucose level in this blood vessel. I think maybe, for example, the higher, not all blood vessels contain the same content of blood glucose, especially after having a meal of rice, meal of carbohydrate, meal of starch. Hepatic portal vein carry more blood glucose from our small intestine towards liver. So this has the highest concentration of glucose. That's much greater than hepatic vein itself, for example. Okay. Now look, difference 180 minus 135, divide 135 as initial times 100 equal this figure. Okay. Look, please, we have to convert to nearest whole number. So only 34% increase. Okay. Let's see this problem coming from paper 4 variant 1, May, June 19, question 2C triple I. Okay. A student used the apparatus shown in figure 2.1 to investigate the effect of temperature on the rate of photosynthesis of leaves of Chinese plantain, Plantago asiatica, at 2 different concentrations of carbon dioxide, A and B. Okay, 2 different concentrations, 370 part per million CO2 and 1000 part per million CO2 at B. Look, please calculate the percentage increase in the rate of photosynthesis at 30 Celsius when the carbon dioxide concentration was increased from A to B, okay, as shown in figure 2.2. Show your working and give your answer to the nearest whole number, okay? Look, please. Also, percentage increase to the final time or last time, final minus initial divide initial times 100. It's okay? Here for at 30 Celsius on x-axis, we extend our ruler to hit A at this point, then hit B at this point, then at A, we have to measure rate of photosynthesis micromole per squared meter per second. This rate, okay? Rate means something over time, look at the figure, I think 22, okay, at this point, then reach 30 micromole per squared meter per second at the second step at B, okay? So there is difference. Now compensate with figures. 30 minus 22 over 22 times 100 equal 36.36. So to the nearest whole number, only we reach this figure, 36%. It's okay? Thank you. Meet you next time, inshallah. I will manage to make more and more revisions for you. May God bless you. Thank you. Bye bye. Bye bye.